So without any further ado then, it is my great pleasure to introduce our first invited speaker. So this is Dr. Melissa Wilson from NHGRI. She's a senior investigator there, studying sex differences in health and disease. Her lab broadly collaborates with clinicians, physiologists, computer scientists to understand the cause and consequences of sex differences in human and non-human animals. At a very personal level, I think I first met you 8-ish years ago, something like that. And she's just a wonderful person. I just got really inspired by her science, and she just has this like really charismatic personality. If you catch her at lunch, maybe you can ask her about her lunch meeting with some notable people at NIH. I'll leave that as a spoiler for her. So it's my great pleasure. We're going to hear about somatic loss of X and Y chromosomes and how that affects tumor gene expression. Thank you, Melissa.
Thank you. All right. I'll start off by saying that if you're a trainee, I noticed a lot, and you're nervous about giving presentations, that never goes away. I noticed that Galaxy was published in fall of 2005, and that's when I started graduate school. So it's still around, and I'm still here, and that's exciting. So, okay. I did just start at NHGRI, so I'm also excited to meet all of the people at NHGRI. I've met many people online, but now we get to be in person. So, okay. So what I want to talk to you about today is maybe one of the coolest things I've ever done. And this is not pre-printed yet. It will be. So if you hate it, just wait a few weeks and you can comment on the preprint. If you have thoughts about it, come talk to me at lunch. If you love it or there's other ideas, right? This is some of these figures were just made last week. So again, it's super cool. Mike also mentioned like calm discussion. I'm not a calm person. I am a very enthusiastic person. So if I ask you many, many, many questions, that's not you. It's just that I can't stop. Okay. So what I have to start with is talking about sex differences. So many people study sex differences, and I have to acknowledge that these are multidimensional. So when people are studying sex differences, they may mean genetics, they may mean specific genes, they may mean sex chromosomes, they may mean gonadal hormones, this whole other axis. They may mean physiology. And then you could imagine this whole thing just like hurtling through space-time because it all changes via development from conception through old age. All of these things do. My lab in particular tries to focus on a really narrow piece of this, which is the human sex chromosomes. They are beautiful and wonderful, and I will note that one of the first QC steps for a lot of studies is to remove them. This is it's a colored image of a human X and Y chromosome, and so just a couple of things to know about them is that they evolved from a pair of non-sex chromosomes, so they used to be indistinguishable from each other. But over the course of several million years, the Y chromosome lost about 90% of the gene content it shared with the X. It also gained some gene content. And for the record, it's not the smallest in the genome. The most gene poor, but it's not actually the smallest chromosome. So there's no reason for excluding it. In concert with the loss of content on the Y was the evolution of something called dosage compensation. And a totally new gene called exist that only exists in the common ancestor of eutherian mammals. And this gene is involved with counting the number of X chromosomes and silencing every X chromosome in excess of 1. So I'll also say that the thing that we care about a lot is understanding the sex chromosome complement in a cell. I love people, but I don't trust any of you. I don't think it's not common to know what your sex chromosome complement is. And moreover, like I said, things can change. So let's just talk about what's typical. Okay, yeah, not calm. Why is the most common genetic variation? The most common genetic variation in the whole genome is the sex chromosome complement. And so it is infuriating to me when they are removed initially. But if we go for it, oh yeah, it's small. That's the small X chromosome. So the inactivated X chromosome is going to be represented there as a slightly smaller one. So this inactivation, we think, leads to roughly equal dosage between XX cells or individuals with XX and individuals with XY sex determination. But more importantly, between the X chromosome and the autosomes, there's actually two things going on there. Cool, well, probably, typically, individuals will have two X's or an X and a Y, but 1 in 2,500 people who are assigned female at birth only have one X chromosome. Sometimes that's because they didn't inherit an X, sometimes that's because they didn't inherit a Y. Could be either. 1 in 500 assigned male at birth individuals has two X chromosomes and a Y. And there's a really cool study, and I forgot to add it in here, of the Million Veterans Project that identifies about 1 in 350 veterans have a sex chromosome aneuploidy, either XXY or XYY. And over 70% of them were not diagnosed past age 60. But individuals in the Million Veterans Project who had a sex chromosome aneuploidy had higher mortality and higher morbidity. Because genetics matter. Okay, and then about 1 in 1,000 assigned male at birth individuals have an X and two Y. About 1 in 1,000, you see the parallel here? Look at the little Y chromosomes and the little X chromosomes. 1 in 1,000 assigned female at birth individuals have 3 X chromosomes. This dosage compensation process allows multiple copies to persist in the population in functional, living, walking, breathing, cool human beings. About 1 in 18,000 assigned male at birth individuals have 2 X's and 2 Y's. And almost about the same, about 1 in 20,000 assigned female at birth individuals have a condition called Sawyer syndrome, where they have an X and a Y chromosome, but develop female gonads. So if we're thinking about AnVIL and Tara and having hundreds of thousands or 200,000 or 8 petabytes of data of individuals, many of the people just typically walking around who are being involved in these projects are going to have sex chromosome aneuploidies. Moreover, you can lose a Y chromosome across life as individuals age. Individuals with a Y chromosome are likely to lose it as they age. In fact, every single one of them, according to studies from Northern Europe, will lose a Y chromosome as you age in a proportion of your cells. But the rate of loss is different. So it's kind of hard to predict. And because I like symmetry and it happens to be true, exist is also lost over life. It's called X erosion. And you can lose an X chromosome or lose, oh yeah, but that's the thing, is that sometimes you lose the X and sometimes you just lose X inactivation. Okay. So how do we get involving AnVIL? Luck. I think luck and that we care about this thing, and I stand on my soapbox all of the time, and every time I raise my hand, you know that I will ask you, did you look at the sex chromosome complement? Did you include the X and the Y? So then they said, well, maybe we should have that voice in the room. And then, I'm like, meh. Then I don't even have to ask the question. I just have to be there. And so we were involved as a use case for understanding sex chromosome complement across these large data sets. We did a collaborative project with AnVIL credits to, I'll show you this preliminary data project. And then we're also, hopefully at the end of the story, going to be doing some more work on AnVIL and single cell data sets. And we were, when I was in academia just a few months ago, part of a collaborative NCPI project led by Benedict Payton. So what we did with this initial study with Mike and Samantha and Brian was look at how does the sex chromosome complement of your reference genome affect your ability to detect variants on the sex chromosomes. And spoiler, a lot. And you will call an additional 4 to 6% mutations, single nucleotide polymorphisms, if you take the sex chromosome complement into account when you're doing your alignment and variant calling. And that project was really useful for us. We learned a lot about, this was led by Brendan Pinto in the lab, with a lot of guidance from Mike's lab and Samantha on developing the little workflows. So, and now for something completely different. We're going to, this project, okay, this is the cool, all right, okay. I both want your expectations to be high and also very low. So we're assessing functional loss of X and Y in 3 data sets. So the typical normal tissue, so unaffected, not cancer tissues are from the Genotype by Tissue Expression Consortium, and you'll hear this data set brought up a lot. It's roughly 950, a little bit, almost 1,000 people, roughly 20,000 samples from 54 regions of the body. So we're using that as our normal data set. And then we're also going to look at functional loss of X and Y in the Cancer Genome Atlas data set. This is around 11,000 patients across 33 tumor types. And then we also look at cancer cell lines. I think roughly a little over 1,000 cancer cell lines from 37 different tumor types. They were derived from different tumor types. And this all started as a course-based undergraduate research experience. So I'm really excited to talk more about the training in AnVIL going forward, because what we had done was we designed a completely asynchronous online research experience to allow students in ASU online to get involved in research in a way that they would never be able to before. And I thought what we would do is let's just look at loss of sex chromosomes in CCLE in the Cancer Cell Line Encyclopedia. And then, oh, I didn't expect that. And then the next question, and then the next question, and then today. Oh no, how do I go back? This is led by Seema Plaisir, a staff scientist I collaborate with, and in collaboration with Ken Buteau, a colleague who was at NCI and is now at Arizona State University. So I want to say, like, how do you assess functional loss of X and Y? So you don't always have DNA for every sample, and what we really care about here is expression. So we're going to look at gene expression, and I'm going to walk you through how we did this. And so it turns out that even though individuals, there's a whole chromosome copy number variation, most of the genes on the X chromosome have indistinguishable expression between XX and XY samples. The only one that really, really is different all of the time is exist. And that's because it's involved in the counting and the silencing. And so in normal GTEx tissue, we can use that. And at the very bottom, the dark blue are reported female samples, and we can see high expression, bright red, right? Exist is expressed there. And reported male samples, dark blue, no exist. Good, because if not, then I need another marker. And then we look, thank you. And then the rest of these are Y chromosome genes. And we did do an analysis of expression of Y chromosome genes across GTEx, but I just want to show you the short arm and the long arm of the Y chromosome. And in general, in reported female samples, there's no expression of the Y-linked genes, and in reported male samples, we do have expression. So then we went to TCGA, and we see some loss of exist there in the reported female samples, and loss of Y chromosome. So this is what we had first done. And we actually, I'm telling you all backwards, because we actually started in the cancer cell lines where you see almost half of the reported female samples have no exist. And there's that one little gray bar at the bottom, which we won't talk about. Those are cell line. I'm telling you about it. Why did I say we're not talking about it? They're unknown samples. So there's no reported sex of those samples. And it turns out from the gene expression, we can infer sex chromosome complement in fewer than half of them, though. So there will be half of these unknown samples that are just completely unknown because they don't have Y expression and they only have expression of a single X. So we don't know the origin of those. Okay, so we did this, but what does it actually look like across tissue types? So when we're thinking about the NCPI, part of this interoperability is how would one as a researcher, and you can't read the bottom lines, but the bottom lines are all the different tissue types in the Genotype by Tissue Expression Consortium. And so we took those in GTEx and we looked at expression. Each point is a dot, is a sample from the GTEx consortium. And we looked at expression of exist and inferred whether the sample had 2X copies or not. And in most of GTEx, in 97% almost, we have expression of exist. And then we tried to line those up one-to-one with the Cancer Genome Atlas data sets. And we see that in the TCGA, over half of the Cancer Genome Atlas data sets have lost typical expression of exist. So then we went back to DNA in TCGA, and we see that that is largely driven by reactivation. The dark purple there is XAXA, which means you have 2 active copies of the X. And then a little over a third of those are complete loss of the X chromosome based on the DNA. So you're seeing both of those things happen. And then finally, in the cell lines, we see almost bimodal, which kind of makes sense. If you start from a single cell and you're growing it out into a cell line, we see either you have expressed or you don't. And so the violin plots, they're not as beautiful as you would like because each tissue type from which a cell line was derived, we only have a handful of representatives. So it's 2, 3, 4 there. Sometimes 1. You'll see like a little dot, but we still put it there because it's data. And in about half of those, sorry, actually a little more. So more often in the cell lines, we see total loss of an X chromosome rather than reactivation, which is not what I expected because I know in induced pluripotent stem cells, you get reactivation of the X chromosome. So I don't know why. I don't know why. Okay, and then we do the same thing with the Y chromosome, and we see that in GTEx, typically individuals have full functional expression of Y chromosome genes. In cancer, it's about a quarter of tumors have lost functional expression of the Y. When we look at the DNA, if you've lost expression of the Y, you have lost the DNA from the Y. There's almost no epigenetic silencing. That does happen, but we don't see it here in the cancer. And then in the cell lines, it's nearly half of the cell lines have lost functional expression of the Y chromosome. Okay, well, so does it matter? Does it matter at all? So then we're going to do differential expression between a typical X active, X inactive cell collected from a female patient compared to, and we're just going to do this in TCGA because that's where we have the power to do it. Call out here, as someone who does also analyze lots of other people's data, previously generated large consortium data, they're often not designed in a way to look at sex differences. And here you wouldn't even know to begin with, to actually collect it, to look at loss of sex chromosomes. So we're limited in what we can do, but we do our best. So we'll compare X naught samples. So you lost an X chromosome from an XX cell, or compared to, so XX versus X naught. And then we'll also compare XY cells to X naught. I apologize. There's a lot of these figures when you're doing big data where you're like, yeah, just squint and look at the trend. Okay, that's what we're doing. So what this is XX samples compared to, with an inactive X compared to XX samples with an active X chromosome. And along the bottom are all the tumor types where we had sufficient sample size to compare. So at least 3 individuals in both cases. And red is upregulation and blue is down. And what you can see is that as you go across, genes that are upregulated when you reactivate the X are consistently upregulated across tumor types. That's all the red there. And genes that are downregulated when you reactivate the X are consistently downregulated across tumor types. Most of these are not on the sex chromosomes, and I'll show that in a minute. And often these genes are involved in cancer. The second bar is yellow. This is a great figure for a paper, less of a great figure for a talk, but you can get the reds. And then I'm going to show you the same thing. So when you lose the X completely, we generally see upregulation across the autosomal genes. So most of the signal and conserved across tumor types is upregulation. And there's a little bit of downregulation when you lose a whole X chromosome. And then the question is, well, what happens when you lose a Y? So you can compare tumor samples that have lost a Y to the same tumor type where you've retained the Y. And we only have 3 tumor types where we have enough to look at it, but it's almost completely downregulation of autosomal genes. And there are 1,500 genes that are consistently downregulated on the autosomes when you lose a Y chromosome. What? Okay, this is cool. Okay, so where are these genes? So the outer one is typical sex chromosome complement compared to reactivation. The middle circle is typical sex chromosome complement compared to loss of X, and the inside is the loss of Y chromosome. They're everywhere. They're everywhere. They're not isolated to one chromosome. They're not only on the sex chromosomes. In fact, the bulk of the signal is on the autosomes. So what does it really mean if we look at the tumors in multidimensional space? So we wanted to ask, how does loss of X or loss of Y affect autosomal gene expression? So we did an ANOVA with the 4 groups. We just do loss of X from an XX, from a predicted XX individual, so reported female patients that have lost an X chromosome, and typical XX samples, and then we did typical XY samples, and then loss of Y. And with those 4 groups, we looked at multiple, so we looked at the top 3,000, the top 1,000, the top 100. They all look very similar, overly honest methods. I'm going to show you the top 1,000 because it looked the nicest. These are so in this multidimensional space, when we do the analysis of variance of the most variable genes across those 4 groups, the first thing that we see is something we see when we're doing RNA-seq in general, which is that XX and XY samples, or more specifically, because not everyone looks at it, male and female samples separate out on the ANOVA. What happens when you lose a sex chromosome? Oh my God, look at it. They're right on top of each other. So then we did differential expression between XX reported female samples and XY reported male samples, and then loss of X from reported female and loss of Y from reported males, and it's almost nothing. There are almost no differentially expressed genes between two. What? The sex differences on autosomal genes are disappearing when you lose a sex chromosome. We've done a lot of QC to try to convince ourselves that we have not made this up. Like we have like double checked and triple checked, and we look at the pipeline and we run it again. Okay, so they're looking more similar, but we care about people, right? I said I care about you. I don't trust you, but I care about you. And so what does it matter for life expectancy? So I'm going to show you the Kaplan-Meier curves, and we start out again with reported male and reported female individuals, but here with the typical sex chromosome complement. And what we know from large-scale studies and what we see here, we have to lump all of the TCGA patients together because we don't have sample size, again, to do it individually. But we see what we expect is that females on average have longer life expectancy. So on average, the median survival for XX female is 12 years. And for XY male is 8, right? 8.8, right? So we know that consistently across nearly all tumor types that males have higher incidence, higher mortality, higher morbidity, worse response to treatment. So what happens when you lose an X or you lose a Y? You lose the sex difference completely, and everyone does worse. Significantly worse. If you just reactivated the X, it doesn't affect survival at all. So losing a whole chromosome has an effect. And here's where the point where then it gets really sad. So then we have to talk about cool new stuff. So what we're hoping to do going forward is to ask some questions. So is that loss of X and Y in these bulk data sets, is that driven by normal cell proportion? Because the dirty secret of cancer genomics is that your tumor sample is not all tumor. Depending on the tumor type, your tumor sample might be 10% tumor, might be 50% tumor, might be 90% tumor. So one thing we can look at if we have single cell data sets, which we would like to do on AnVIL, is to look across and see, is this heterogeneity being driven by normal cell proportion? Is it being driven by cell type? So are we losing the X or the Y in a particular cell type that might be causing this? Or is it just regional heterogeneity and expansion across as tumors grow? And we know that we can have these like branches of somatic variation that exist in a tumor. So that's where we would like to go. And hopefully we'll find something cool. And hopefully, if you take anything away, it's that you should trust no one and look at the sex chromosome complement of your sample. So thank you to everyone who is a part of this and to our previous funding before we moved to the National Human Genome Research Institute. And most importantly, thank you all for being here. Oh, and I am legally obligated to say that I am now at the forefront of genomics. So, yeah. Questions? One and then two. Yeah. I was going to ask. Oh, yes. Sorry. Oh, wait. Here.
I mean, I can shout, but that isn't helpful for Zoom. Yes. Okay, so what I was wondering about is what the different future directions were going to look like in a little bit more detail. But that might be a longer question, so maybe do the other one first.
Yeah, I mean, I think part of this is working with more people who are generating their own data sets, who are working with their own clinical records, who are working with their own patients to see not only in cancer, but in, I know there's really great work coming out in COPD, in other aging related diseases, in Alzheimer's and dementias, in many conditions that loss of sex chromosomes might be important. And I want to say, like, I don't say we should only look at the sex chromosomes. It's just that we should look at the whole genome. In the way that we talk about the T to T, let's look at the whole genome. Then don't leave 5% of it out. Yeah. I have to earn my lunch.
Thank you, Melissa. Great talk. I have a question on for the GWAS end. My personal experience is, at least on the array, a lot of times chromosome Y will have really high missing rate. I'm not saying that's why they're excluded, but most of the time chromosome Y is not used in GWAS. And from my limited personal experience, a lot of times Y chromosome is just much higher missing rate than any other chromosome in the genome. Do you think there's any reason why that's the case that really hinders GWAS to move to more sex chromosomes?
Do you mean the Y is missing as in it's been lost and so there's no variation called on it, or it's not incorporated, or?
From my experience, if you just look at the marker missing rate, Y chromosomes have 10, 20%, which is really high for array.
Yeah, I think many things. I think one of the things I didn't point out, but one of the students found, is that in a subset of samples from CCLE, individual samples had lost only the P or only the Q arm. And so if we also know that samples can lose a Y as they age, then in GWAS or other association studies, you might not have variants there to look at. And I don't know how we incorporate loss of a region, so loss of genes, into a GWAS analysis. It sounds like room for more methodological approaches.
Yeah, and this sort of, sorry, I'm taking up a lot of time. This sort of goes into some imputation problems we run into. Chromosome Y is basically never imputed. You can't impute on something that's completely missing, essentially. But yeah.
Well, so I wonder, I'm thinking off the cuff here, but the HPRC, there's a Y chromosome subgroup analysis, and the way that the Y evolves, it's a single non-recombining unit other than the pseudoautosomal regions. And so I wonder if there might be ways based on predicted geographic ancestry of the sample to try to impute, but maybe moreover, it's not that you need to impute something that's not there. Maybe it's important that part of it is not there. I mean, it just gets messy, right? Like, that's the thing that's wonderful and interesting, but it's, ugh.
Thank you.
I'm happy to point out problems and then let you all solve them.
Emma, that was great. I have a question too. Did you look at the overall, like when you have these loss and their worse outcomes, is there any other, are those, does that correlate with like pure other loss, like pure other variation, like pure variation burden? Are these people just like way up there in variation burden generally?
So and that's a great question. And it's true that we did look at predictors of aneuploidy in general across the genome, and high aneuploid tumors are more likely to have lost the X or the Y. So they're really confounded variables there. And we we barely had the sample size looking across all of TCGA, so trying to disentangle that. What I can say is when we did the same analysis, like survival analysis across every single chromosome, we didn't always see the typical expectation of samples from reported females and reported males with, let's say, 2 copies of chromosome 1 being in the same direction as one would expect. So I don't know how to interpret it yet, other than, yes, individuals with high aneuploidy are going to have worse prognosis, period. And then you're more likely to lose an X or a Y if you have high aneuploidy. Yeah.
Hey, Melissa, it's really great research. I'm just curious about the other species. Is there any other species that lose XY chromosome? And if there's any behavior change or behavior patterns change on the sex chromosome employees? Thank you.
Yeah, the X and Y chromosome system that we have is shared across all eutherian mammals. And so everything other than the marsupials, which other than the North American opossum are all in Australia, and the monotremes, the egg-laying mammals. So they do. As far as how often does aneuploidy occur across those populations, I think we don't even have reference genomes for most of those species, let alone population variation for it. So I imagine it's there, but I don't know off the top of my head the estimates.
So I have about 1 million questions.
Ooh. That
this
PS, that
happens.
clock is like 2 minutes fast. You have time for one.
Okay. About how this happens, implications. But since we're at the AnVIL Community Conference, I'll just ask one. How else can AnVIL help you?
Oh, well, there's like 1,000 ways, but wouldn't it be cool to like incorporate sex-aware inference into general pipelines in AnVIL that everybody does, just so that when they do their point and click, like I'm going to run my Galaxy pipeline, I'm going to run my Woodle pipeline for variant calling. I'm going to do it in a way that's aware of the sex chromosomes and gives me an inference for my sample. And at least then, even if you're going to exclude it, at least you have it there and other people could look at it.
Let's do it.
Yeah.
All right, let's thank Melissa here again.